Hey guys, welcome back to One Stop Biology. So today we are going to discuss various topics, which includes the search for genetic material and how basically we got confirmed that the genetic material is nothing but the DNA, right? So in the previous video, we studied all about DNA and what is its composition, how is it made up of, what are the different type of linkages which are present, what are the different type of compositions of DNA, right? So in this video, we are going to study all about the confirmation that we got, ultimately that DNA is the genetic material and nothing else. So before we start off, guys, please do not forget to give feedback on this video after you watch it completely. Please do not forget to like it if you did understood the video. Please do not forget to give your comments. Either you like it or if you have any doubt, right? So I will be waiting for your comments, guys. So basically, what happened is that after the discovery of Mendel, which was principles of inheritance, almost at the same time, people started working on the genetic material, which was first considered as factors, then genes, right? So in 1926, basically this quest to determine the mechanism of this gene got proven, and it was again and again proven by various scientists. And in 1928, the transforming principle came into existence. Now, what is this transforming principle? So, basically, what happened is Frederick Griffith, it is also known as Griffith Experiment, guys. So, Frederick Griffith in 1928, in a series of experiment with Streptococcus pneumoniae, with Streptococcus pneumoniae, witnessed a miraculous transformation in bacteria. So basically, he experimented on Streptococcus and he witnessed some transformation. Now, what was that transformation? So when Streptococcus pneumoniae bacteria are grown on culture plate, they produced smooth, shiny colonies, right? So basically, when these bacteria were cultured in on a plate, there were some who made smooth, shiny colonies, and then there were some who made rough colonies. Okay, this was S and this was R. Now, this happened because the smooth strain bacteria have a mucus coat, right? They have a polysaccharide coat, while the rough bacteria, the R strain, they do not have any such coat, right? Now, what he did is he infected mice with smooth strain first. Now, guys, remember that the smooth strain was the virulent phase strain, right? So he infected mice with the S strain, which was the virulent strain, and the mice died from pneumonia infection. But when these mice were infected with the rough strain, they do not develop pneumonia and they did not die, right? Now, basically, Griffith was able to kill bacteria by heating them. What he did is that he saw that you can heat this bacteria and these bacteria will get killed because of high temperature, right? Now, what he observed the next, that he that heat killed S strain, the heat killed virulent strain, was when injected in mice, the mice did not die, right? Because the bacteria was killed, right? But when he injected a mixture of heat killed and live R strain, which was non-virulent, when this mixture was injected into mice, the mice died, right? Ideally, the mice would not have died, right? Because this already is heat killed and this is non-virulent. But the mice died. So what he concluded? He concluded that the non-virulent R strain, the rough strain bacteria, somehow, remember, somehow this bacteria was transformed by the heat-killed S strain. So that means in this mixture, somehow this was transformed by this, right? And that enabled the R strain to synthesize a smooth polysaccharide code, and that became virulent. And because of this, the mice died. Now, this must be because of the transfer of genetic material. This must be because of the transfer of genetic material. But what happened was that the biochemical nature of this genetic material was not defined from his experiment. So he basically predicted something, he concluded something, but that was not inferred, right? So he said this might be due to the transfer of genetic material. Now let's see what was the biochemical characterization of transforming principle. So basically, prior to the work of Oswald, Avery, MacLeod, and McCarthy, the genetic material was thought to be a protein. People said that the genetic material was a protein. Now, these three, Oswald Avery, Colin MacLeod, and Maclean McCarthy, in 1933 to 1944, so for almost 10, 11 years, right? So, from 1933 to 1944, they worked to determine the biochemical nature of transforming principle, which was done in Griffith's experiment. So what they did is they purified the biochemicals, the proteins, DNAs, and RNAs from the heat-killed S strains to see what what basically transformed into the R cells, right? Now they discovered that DNA alone from the virulent strain, the S strain, caused the non-virulent R bacteria to become transformed. So DNA alone was responsible. They also discovered that protein digesting enzymes, which are proteases, and RNA digesting enzymes, which are RNAses, did not affect the transformation. So these protein and RNA digesting enzymes, they did not affect transformation. So the transforming substance was not a protein or RNA, right? So basically, DNA was the hereditary material which made the non-virulent R strain into a virulent strain, a virulent S strain. So this was the biochemical trans characterization of the transforming principle. That is how we concluded that the genetic material is nothing but the DNA. Now, this was again proven by Alfred Hershey and Martha Chase. The experiment is known as Hershey and Chase experiment. And basically, this Hershey and Chase experiment, this was done in 1952. What they did is that they worked with viruses that infect bacteria. These are known as bacteriophages. Right? So they worked with bacteriophages. Now, what happens is that the bacteriophage attaches to the bacteria and it transfers its genetic material into the bacterial cell. Right? Now, this bacterial cell treats viral genetic cell as if it was its own. Right? So the genetic material which is basically transferred into the bacteria becomes the genetic material of the bacterial cell, and those manifests the bacteria. And once the bacteria basically agrees that the viral DNA is of its own, it starts manufacturing more virus particles, more genetic particles from the virus. Right. So Hershey and Chase basically worked to discover whether it was protein or DNA from the virus that enters the bacteria. So what they did is they grew some viruses in a medium that contained radioactive phosphorus. Okay, let me just show it through the diagram itself. Yes, this was the Hershey and Chase experiment. So what they did is they grew the viruses on a medium that contained radioactive phosphorus. Okay. Now, what happened is there was some other medium. So basically, they used medium that contained radioactive phosphorus and then some other virus. So some viruses here and then some viruses in radioactive sulfur. Okay. Now, what happened is that the viruses grown in the presence of radioactive phosphorus contained radioactive DNA, but not radioactive protein, because DNA contains phosphorus, but protein does not. Right? You have phosphorus in DNA. I hope you remember the structure that we discussed in the last video. If not, please go through the last video once. Now, similarly, the viruses grown on radioactive sulfur, they contained radioactive protein, but they do not, but they basically they does not have radioactive DNA. Why? Because DNA does not have sulfur, while protein does. Right? Now, these radioactive phages were allowed to attach themselves to E. coli. So here, if you can see, they got attached to E. coli. Right? Now what happened? So after they infected the E. coli, the bacteria, the viral coats were removed from the bacteria and the virus particles were separated from bacteria by spinning them in a centrifuge. Now bacteria which was infected with viruses that had radioactive DNA, which means radioactive phosphorus, here, this one, because this has radioactive phosphorus, this has radioactive sulfur. So the bacteria which were infected with DNA, with radioactive DNA, indicating that DNA was the material. Basically, it was radioactive, right? DNA was the material that passed from virus to bacteria. So here the DNA passed. Now the bacteria that were infected with viruses that has radioactive proteins, that means that has radioactive sulfur, were not radioactive. There were no radioactive bacteria. Here there were radioactive bacteria. Right? So this basically indicates that proteins do not enter the bacteria from the viruses, but DNA does. But DNA does. Right? So basically, after this experiment, after the Hershey-Chase experiment, it was finally proven that DNA was the genetic material that was passed from the virus to the bacteria, and DNA is the genetic material that we always talk about. Now, what are the properties of the genetic material? Let's see that, right? So, a molecule that can act genetic materials must fulfill some of the criteria, right? So, what happens is that the first criteria is that it should be able to generate its replica. So the first criteria is that it should be able to perform replication. It should be able to replicate itself, right? The second property is it should be stable both chemically and structurally. So it should be a very stable structure and it should have a very stable chemical composition, right? Now, the third point is that it should provide the scope for slow changes that are required for evolution. So, guys, remember that mutation, any kind of mutation or slow change, is not necessarily, bad. Sometimes, because of evolution, because of slow changing environment across us, our body keeps on evolving, which is good for our survival, which is essential or mandatory for our survival, right? So that we better, we have a better living. And that is again very important. So the DNA or the genetic material should be able to provide scope for slow changes. Now, these slow changes are required for evolution. So, this was important. Now, the fourth point, again very important, that it should be able to express itself in the form of Mendelian characters. So it should be able to express itself in the form of the Mendelian characters that we studied. It should be phonetically or physiologically be expressed, right? So these are the four rules that were made for genetic material to, for a molecule that has to be a genetic material, right? So now because of rule of base pairing and complementarity, both DNA and RNA have the ability to direct their duplications, right? Now, the other molecules in the living system, such as proteins, fail to fulfill the first criteria. So, DNA, yes, they fulfill the first criteria. RNA, yes, they fulfill the first criteria. But proteins, no, they do not fulfill the first criteria, right? Now, the genetic material should be stable enough not to change with different stages of life cycle, age, or if it's a change in physiology of the organism, right? So they should be stable chemically and structurally. Now, stability as one of the properties of genetic material was very evident in Griffith's transforming principle, right? That heat which killed the bacteria at least did not destroy some of the properties of genetic material, right? So this now can easily be explained in the light of DNA, that two strands being complementary, if separated by heating, if they come together when appropriate conditions are provided, right? Now further, if we see that the OH group present in nucleide in or RNA, so we studied that in RNA at second carbon we have OH group, yeah, right? That is a reactive group and it makes RNA labile and easily degradable, right? So RNA is now known to be catalytic, hence reactive, right? Now here what we can say that DNA then chemically is more reactive and structurally more stable when compared to RNA, right? Now that is why we say that among the two nucleic acids DNA and RNA, DNA is the better genetic material. Remember that DNA is a better genetic material, right? There is one more factor that the presence of thymine at the place of uracil in case of DNA gives DNA extra stability. Remember that. right? Now, both DNA and RNA are able to mutate. They are able to show changes, right? All, in fact, we say that RNA is even more unstable than DNA and it basically can mutate at a faster rate. That is why viruses having RNA genome have shorter lifespan, mutate and evolve faster. So both DNA and RNA can evolve, right? Now, so third point again is fine, right? Now, RNA basically directly codes for the synthesis of protein. So they can easily express the characters. DNA, however, is dependent on RNA for synthesis of protein. So basically, how does a protein synthesis happen? It happens from DNA to RNA and then protein. So here, basically, RNA directly codes for protein, but DNA is dependent on RNA to code protein, right? So the protein synthesizing machinery has evolved around RNA. Remember that RNA is very important. But all the above points, if we consider, we can say that RNA and DNA both can function as genetic material, but DNA being more stable is preferred for storage of genetic information, right? And for transmission of genetic information, RNA is better, right? Now let's see what is the RNA world. Till now we have discussed what is DNA, right? Let's see what is the RNA world. What is RNA all about? So basically, RNA was the first genetic material. Remember that RNA was the first genetic material. Now, if we talk about metabolism, if we talk about translation, if we talk about splicing, all these processes evolves around RNA. So RNA basically is a genetic material. It also acts as a catalyst. So it is a genetic material as well as a catalyst. Now, but there's one consequence as well. Since it is a catalyst, it is considered as more reactive and hence is unstable, right? So since RNA is more reactive, it is unstable as well, right? Therefore, DNA has evolved from RNA with chemical modifications that makes it more stable. And DNA is more stable because it has evolved from RNA with chemical modifications, right? Not DNA, there is also one thing with DNA that DNA has double strand, right? DNA is double stranded and it has complementary strands. Now, these properties resist changes in DNA as what happens is they repair itself. If there is any anomaly, what they do is they repair itself and that makes DNA more stable. But RNA being the first genetic material, there are a lot of important processes like translation, like formation of proteins, which evolves around RNA, right? So guys, we have already covered what was the basis of the proof that DNA itself was a genetic material and what basically RNA is, right? What is the difference between a DNA and RNA, and what are the points to remember if a molecule has to be the genetic material. So with this, we have finished this video, and in the next video, we are going to study replication. Now, I hope you have understood the previous video and this video where we discussed all things, all important points about DNA and RNA. Please do let me know if you like the video or if you have any doubt. Please do not forget to leave a comment on the video and do like the video, share it with your friends. And if you are new, please do not forget to subscribe to the channel. Thanks, guys. Bye bye.